welcome everybody. My name's Dan Hanley. I'm one of the members of the Trial Innovation Network, which is an NCATS program for multi-institutional program support and development in the area of multi-site randomized clinical trials. This is going to be the first in our series of symposia that are going to be semi-annual approximately, and entitled DIDACT, taking its name from the meaning of didact, to teach and to learn, which was a program started by the Johns Hopkins CTSA in about 2015. And we're re-energizing that program and are very excited to have your participation today. The idea behind the program is to share expertise and some of the resources that expertise has developed in this area of multi-site randomized clinical trials. Our program you know about today, so I'm not going to say any more about it. This is the, thank you, Cecilia. This is the Trial Innovation Network as it exists for the 2023 to 2028 time period. And you can see all of the institutions that are partnering with the entire network, and specifically with the Vanderbilt Recruitment Innovation Center, the Vanderbilt Trial Innovation Center, and the Johns Hopkins Trial Innovation Center. And we're attempting to expand expertise and inclusivity, and you can see some of the areas that we're targeting, and DIDACT is one of our mechanisms of supporting the consortia. I think that's we have one more slide, the agenda slide. Yeah. So Guyan, will you introduce yourself? Guyan will co-host this with me. Guyan?
Of course. And welcome everyone. It's great to be here. My name is Guyan Yanokian. I'm a biostatistician and a faculty member at the Johns Hopkins Bloomberg School of Public Health. I am also part of the Trial Innovation Network, and I co-lead the Johns Hopkins BIRD program, the Biostatistics Epidemiology Research Design.
Thank you, Guyan. And Guyan and I see this program as a resource for all CTSA individuals in the work they do in developing the local interest in both single site and multi-site trials at their sites. So Guyan, would you like to introduce our first speakers?
Of course. It's my pleasure to introduce Dr. Michael Rosenblum, and who is a professor in the Department of Biostatistics, Bloomberg School of Public Health. And he's going to talk about the improving precision in estimating marginal treatment effects in randomized trial. Michael?
Thank you, Guyan, and thank you to the organizers. I'm Michael Rosenblum, and my colleague Josh Betz is here as well. And I want to mention another important colleague who's collaborated on this work, Kelly Van Lanker at University of Ghent. I'll be focusing on one technique for analyzing data in the primary analysis, efficacy analysis in a clinical trial. And the goal is it's in the title, to improving precision in estimating marginal treatment effects in randomized trials. A disclaimer, these opinions are just of the authors and not representing our university, the FDA, or anyone else. I mentioned FDA because their name will come up, and some of this work is funded by them, but this does not represent any their views. The FDA put out a guidance document on covariate adjustment. It came out in May of last year, and it's called Adjusting for Covariates and Randomized Clinical Trials for Drugs and Biological Products. It's a very well-written, clear guidance document. I recommend to take a look at it if you have interest. And I'll give you a quote from it. I think the most important quote, if you had to pick one, or just 2 sentences, it says, covariate adjustment leads to efficiency gains when the covariates are prognostic for the outcome of interest in the trial. And then in bold, I boldface this. Therefore, FDA recommends that sponsors adjust for covariates that are anticipated to be most strongly associated with the outcome of interest. And it puts a big smile on my face to see this, because I'm very passionate about covariate adjustment, and I'll try to explain why, and how to do it, some software tools for it. And I also am honored to be some of my work is referenced in this guidance, as well as Dr. Hanley. He's on a paper with me on some of the stuff that is cited in that guidance. So what is covariate adjustment? I haven't explained it. And the setting here is within a randomized trial. And you can do this in the context we'll focus on is just a standard randomized trial where there's a treatment and control, and participants are randomized 50-50 to each arm, though it can be used in other types of trial designs as well. And so these are the 4 key takeaways about covariate adjustment. It's a statistical method for analyzing the data at the end of the trial, where the goal is trying to estimate the average treatment effect. And it has to be, the first bullet point is it has to be pre-planned. This has to be built into the protocol, into the statistical analysis plan that's decided before the trial starts. And the idea is you're pre-planning that there will be an adjustment done for baseline variables, that is variables measured before randomization, when estimating the average treatment effect in the primary efficacy analysis. Efficacy analysis is to see what is the benefit, if any, of the treatment compared to control. And the second bullet point here is that the interpretation of the estimated treatment effect is the same as when you use a simple unadjusted estimator. So in other words, the interpretation doesn't change when using this statistical method versus just an unadjusted estimator. Unadjusted estimator ignores baseline variables. It just compares, say, the mean outcome in the treatment arm to the mean outcome in the control arm, and does, say, the difference or some other contrast. So so we're trying to get the same thing. The average treatment effect is, we're trying to estimate what would happen if everyone in the study population had been assigned a treatment versus had everyone been assigned a control. On average, what's the difference? And the potential benefit, bullet point 3, is that the covariate adjustment method, it can improve precision and reduce the required sample size to achieve a desired power. Precision is how much error there is in estimating the treatment effect. So you can see it, you can picture it in the form of a confidence interval. So at the end of the study, there'll be a point estimate for the treatment effect, and then typically a confidence interval, plus or minus something. And the more precision means a tighter confidence interval. Yeah. And the fourth bullet point is that you can use covariate adjustment for many types of outcomes. So if your primary outcome in your trial is continuous valued, binary valued, ordinal, time to event, I'd say most types of outcomes in clinical trials, there are covariate adjusted methods that give you these potential benefits that can be applied. And the reason for my excitement and passion about covariate adjustment, it's that there's been some confusion out there about when it can be used, how to do it, what's the purpose of it. And so I'll just quickly go through a couple surveys that have been done of randomized clinical trial reports. So Pocock in 2002, 22 years ago, surveyed 50 randomized clinical trial reports, and the finding in this quote is, the statistical emphasis on covariate adjustment is quite complex and often poorly understood, and there remains confusion as to what is an appropriate statistical strategy. In 2010, there was a similar survey and a similar conclusion. And in some ways, this has been, now we're at a very improved state partially because the FDA put out this guidance document that lays a lot of it out there. And, the summary is that they recommend to do it, to adjust for covariates that are anticipated to be strongly associated with the outcome of interest. So that's quite nice. We're out of this confused state into, thanks to FDA's guidance document. And they're similar in European Medicines Agency has a similar, not exactly the same, but pretty similar guidance. And the goal of covariate adjustment I'll be talking about estimating marginal treatment effects, and that's in contrast to something called the conditional treatment effect, which is also something that can be done. I mean, covariate adjustment can be used for that as well, but I won't be discussing that. So here, the target of inference, I already told this, it's the intention to treat estimate. What if everyone had been assigned to treatment versus everyone assigned to control? And then you measured everyone's outcomes in those two hypothetical scenarios and did some contrast, the difference between means, the difference between proportions. It could be some difference in survival probabilities if it's a time to event outcome. And yeah, I think I covered all this already. There's a large amount of related work on this. I did not invent this in any way. This has been around for a long time, even before the earliest citation I have here of Yang and Ciatis in 2001. There's prior work on it in the survey sampling literature. I'll skip some of this and just talk about a couple examples to highlight the high-level ideas. But I hope you ask questions and put questions in the chat or in the Q&A about this. And actually, I want to also ask you if you have questions even unrelated to what I'm talking about, but you have a burning question about clinical trials that you'd like someone to work on, a research question you'd like us to work on, please also put that in the Q&A. We might not answer it now, but I'd love to know just what are the needs? What are the most pressing needs? Because that will help steer future research. Okay, back to this example. So this is an example of planning an Alzheimer's disease trial, which was conducted, but this is before it was conducted. I was on the team designing it, and it was comparing a new drug treatment versus placebo control. And we used data from a previous trial with the same patient population, patients with mild cognitive impairment. And we used data from the previous trial to help plan the future trial. And one thing we looked at is, should we use covariate adjustment and try to, what would be the potential benefit of covariate adjustment in this future trial? And there were three sets of variables that we looked at. The first is the primary outcome. That's the first bullet point here, which is the change in clinical dementia rating sum of boxes score. It's a combination of functional and cognitive outcome disability in comparing 1.5 years in versus baseline. So that's the primary outcome. There's study arms, that's denoted by A, that's a new drug versus placebo, A is 1 or A is 0. And then baseline variables, that's a whole vector, that's a bunch of variables. So you have participant age, gender, and at baseline, how severe was their impairment. So there's different ways to measure that: Alzheimer's disease assessment scale, ADAS-Cog score, mini mental state exam score, some other scores here. And the primary outcome is a change in CDR sum of boxes. And then the baseline variable, we include that value measured at baseline. All of these we picked them because in the literature on Alzheimer's disease, it's known that these are prognostic for the outcome. These are correlated potentially with the outcome. If you start off worse off, that can impact how likely you are should change by different amounts, regardless of what treatment you get. And then we did a simulation study to look at, well, what if you adjust, if you do an analysis where you adjust for chance imbalances, that's the idea, adjust for chance imbalances in these baseline variables between treatment and control arms, to estimate the average treatment effect. And we estimated there's a 23% reduction in the sample size you need to have the same power, comparing this covariate adjusted estimator versus an unadjusted estimator. Unadjusted is kind of the default, I'd say, estimator. So you get it, that's a pretty substantial improvement, and at no cost. That's the other thing. It doesn't cost anything extra to do this as your primary analysis. And then in the real trial, so you can see later what actually happens, and we got a similar estimated benefit from covariate adjustment. And I'll give another example. Let me just cut to another example of a binary outcome. So this is connected to, this is work with Dan Hanley, who is organizing all of this, one of the organizers. And so this is work from a while back that he and I collaborated on, and others. But he's the PI for a trial called the MISTY, minimally invasive surgery trial for treating intracerebral hemorrhage. And there, the primary outcome is the modified Rankin scale, which is a scale from 0 to 6, measuring degree of disability, and measured 180 days from enrollment. And here it was dichotomized as being less or equal to 3. So it's a good outcome if the value is less or equal to 3. That's the cutoff of being able to walk unassisted or not. And a bad outcome if the value is larger than 3. And the study arms, it was this MISTY surgical intervention versus standard of care. And the baseline variables that we looked at for potentially adjusting for, so this is in using previous data to plan this phase 3 trial. And there were a few variables, NIH stroke scale, clot volume, and clot location, that in the prior data set that we had on the same population, they were strongly prognostic for the outcome. And we did something similar, simulation studies based on the earlier data, and we found that you get a substantial precision gain by adjusting for these baseline variables at the end of the trial. You can also use it in interim analyses. And it was equivalent to approximately needing 28% less sample size to achieve the same power, just by using this improved statistical method. And in the real trial, we looked at, okay, well, how much precision gain did we get? You can estimate that. And it was pretty similar, something like 25%. I will say that stroke trials are probably the best case scenario, at least based on my personal experience, in terms of how much improvement one can get. The improvement depends on how prognostic the baseline variables are. The more strongly prognostic, the more of a precision gain is possible. If your baseline variables are not prognostic at all, then this cannot help you. But the way to find out is look at previous data or similar data, same population and same outcome and baseline variables. And in many other disease areas where I've looked at clinical trial data and estimated, is there a benefit or not? Typically there is a benefit, but not always. Sometimes it doesn't, there's just no effect. Let's see, just for time, because I want to make sure to leave time for questions. So some of these slides I'll skip over, but I'll just tell the main point is that you can use covariate adjustment more or less regardless of the outcome type, at least if your outcome is one of the standard ones of a continuous valued, binary valued, ordinal. Ordinal would be a scale, 0, 1, 2, 3, 4, 5, 6. But time to event outcome, the best survival time. And let's see, one of our papers we did looked at COVID treatment trials and what is the potential benefit or limitation of covariate adjustment in that context. And this was done right at the beginning of the COVID pandemic and contributed some ideas to an FDA, we submitted ideas that were later, similar things were in an FDA guidance document on designing and analyzing trials for COVID, where they recommended to apply covariate adjustment because there are well-known baseline variables that are correlated with the outcome. There are certain risk factors, age, for example, that are prognostic for having worse outcomes. So let me see. I want to talk about resources that are available. If you want to apply covariate adjustment, feel free to reach out to me, of course, with any questions. If after this, after today's presentations, if you have any questions, you can just email me any questions. I would love to hear from you the questions, or if you have some need that's an unmet need, I would love to hear that as well. But some resources that are available that we have been working on. So one is this, this is available at covariateadjustment.github.io. And this is created by Josh Betz. I mean, I'm involved, but Josh Betz is the brain behind and the brawn behind all of this. So he gets the credit. And so this is some things that are available, tutorials, online tutorials that walk through how to apply this for real trial data sets, and walk through and show what happens. There's example data sets, code results, and some guidance. And then over here on the right half of the screen, there are some software packages, and these are not made by us, but these are some existing software packages that carry out a covariate adjusted analysis. And the point of this table is that no matter what your outcome type is, there are covariate adjusted estimators that have potential to improve precision. And we have a software tool here that it can use any of those as an input. So we have this general software tool that you can pick what your estimator is from that list or otherwise, and it's to help plan. We call it it's a kind of data adaptive design where the timing of it has interim analyses as in a group sequential design, but the time points where interim analysis takes place, that is computed in a way where it takes into account how prognostic the baseline variables end up being in your study. So it adapts to how prognostic they are, because you may not know how prognostic they will be. And you generally won't, you can't exactly know what that will be. So it adapts to that to have the right power. And so there's some sample size planning, and this is a plot from it that shows, it does a comparison of what the timing will be of interim analyses in terms of how many people have to complete the trial, depending on how prognostic your baseline variables are. And you can combine this with stratified randomization. Often that comes up as a question. It's a really good question. So we have a paper. This one's in, I should have updated this, but it's in Journal of American Statistical Association, led by Bin-Kai Wang, who was my former PhD student. Now he's a assistant professor at Michigan, and he and other colleagues, where we show how to combine the benefits of stratified randomization and covariate adjustment. The ideal thing is to use both together. And then we just have some general recommendations on covariate adjustment, how to compute confidence intervals when you're using covariate adjustment. You have to make sure you're using the correct method that takes into account that you did covariate adjustment. You can combine it. My view is that the best use of covariate adjustment is when you combine it with information monitoring, which means monitoring how quickly information is accruing in your trial in terms of it. That could be proportional to the number of outcomes that are measured or the number of events in a time to event trial. But you can also take into account baseline variable information into that. And so our software tool tries to help with that, in planning trials like that. And there's also missing data. One typically has missing data in clinical trials. And so if we have proposed methods that to handle missing data, which they're essentially the similar methods that you would use if you weren't adjusting for baseline variables, but it naturally adds on when you have baseline variables you're adjusting for. And here are the bottom line pros and cons of using covariate adjustment. So there's potential to improve precision, which you could either convert into smaller sample size or larger power. This third bullet point is one of my favorites, that one might think that in very large trials with large sample sizes, thousands of participants, for example, that covariate adjustment might not be very useful because things should balance out. There's less likely to be large imbalances in baseline variables between the study arms. And I mean, that is true. Things will balance better on average between the study arms at larger sample sizes. But covariate adjustment, I would argue, is even more useful in large trials in the sense that the absolute benefit, the sample size reduction you get from covariate adjustment is larger in large trials. So if you get a 10% reduction, it's the same percentage reduction, at least asymptotically, at large trials and smaller or medium-sized trials. So a 10% reduction in a 1,000-person trial, that's more. That 100 people versus in a 100-person trial, 10% is saves 10 people. You still always want to do the smallest trial, so it's still always, I think, useful. But it's just that don't forget the large trials. That's where actually you get in a sense, the biggest bang for the buck out of it. But I should mention there is a con. I haven't mentioned this yet, but if you adjust for baseline variables that are useless, that are not prognostic for the outcome, they just end up, you thought they were going to be correlated with the outcome, but they're not. Then there's potential to lose efficiency if you adjust for some just noise, essentially. Then you essentially every variable you adjust for uses up, it wastes a degree of freedom. It's like throwing away one data point, roughly speaking. So it is. So I would be. So that downside is less of a downside as the sample size is large in larger trials. And the other caveat, the other thing to keep in mind is that you have to have a minimum sample size for this to work. Typically, this is going to involve fitting some regression models, even though we don't make any regression model assumptions, but you might fit a logistic regression model as part of this procedure, for example, and there you need some minimum sample size to do that. So you can't do this with 5 or 10 person tiny study, but you need some minimum, say 100-person trial that it will, and the number of variables you adjust for, that depends on how big your sample size is. The larger sample size, the more variables you can potentially adjust for. And so I'll stop there, but at this website, we have papers on covariate adjustment, some links to trial planning tools, an FDA short course video recording that I did a while back, and some other information. But please feel free to reach out with any questions, and I'll stop there and let's turn to some questions. Let me just
Michael, there is one question related to baseline covariates. You mentioned that these covariates have to be strong predictors of outcome, and you also mentioned about imbalance between treatment arms. So there is a question about when would you see a benefit in terms of adjustment for this covariate? Is it when the covariates become balanced, or how would you think about this adjustment?
Yeah, that's a great question. And please continue to put in questions while I'm talking here. It's more interesting when there's some back and forth. And so it's a good question. And so typically there will be some imbalance between the study arms and baseline variables. I mean, it's more or less impossible to get perfect balance. You can try to get some balance by design by using stratified randomization, which is generally a good idea. So for example, you can take in the stroke trial example, you could take people who have high versus low severity, stroke severity at baseline, and separately do permuted block randomization in those two groups, and that will give you almost perfect balance on those variables. But you can typically only do that on a small number of variables, and they have to be essentially just a few levels, maybe about 2 levels, or maybe a little bit more than that. So you can try to use stratified randomization to balance what you can, but then there may be a lot left over of imbalance, and that's a random process. And the idea of covariate adjustment is to whatever's left over, it will account for that, and squeeze out noise that's due to that, the imbalance. And so in general, the overall impact of covariate adjustment will be to shrink the confidence interval, and that's it's not guaranteed to happen, but generally will happen if you have moderate to strongly prognostic baseline variables. And that's going to happen regardless of how the actual imbalance in the data, the confidence interval, it'll depend on the data, but the precision gain is it's not contingent on the imbalance in the data. It's in terms of the average effect and the average reduction in the variance. So I guess bottom line for that question is that the covariate adjustment works even when you don't do anything to try to balance the baseline variables between study arms. You have to do standard randomization, but also can work when you do enforce imbalance. And the biggest benefit will be in cases where you have a big imbalance in an important prognostic variable. I mean, that's where it really is going to have a big impact on the pushing things on average in a better direction.
Great, thank you.
Let's see.
There's
We
also
go
question-
more,
Oh,
Diane.
yes, I was going to say that there is a question about power analysis and sample size determination. And so the question is about, do you consider this reasonable to attempt to base sample size on adjusted analysis?
Also a great question of, it's a really hard question. I mean, if you might not be surprised, my answer is it depends. In certain, I mean, I'd say if you have solid evidence from previous trials in the same population that and the same outcome measurement, same baseline variable measurements, that you have strong evidence that the baseline variables will be prognostic, and you have some sense of how prognostic, then yeah, I would factor that in. And the one example I know of is in stroke trials and ischemic stroke trials. There's some work where they I'll have to look up who the authors were of this paper, but it's very well done, where they took, I think it's 6 or 7 completed trials and looked at what is the precision gain in adjusting for some baseline variables in these completed ischemic stroke trials. And they found it's about 25% across the board. So if you're going to do another one of those trials in a similar population, similar outcome, you might say, well, I expect to get some benefit from covariate adjustment. Probably not exactly what I saw in the previous trials, but you might say, let's at least build in some fraction of that, assume that we'll get that benefit, and base our sample size off of that. But in general, it's I would caution against assuming from the get-go that the trial you're planning is going to be the same as a previous trial. I mean, it may be similar, but there's always going to be differences. So, and you don't want to bank on something being the same, such as how prognostic the baseline variables are, when it might be somewhat different. And so what you can do is hope for the best, but plan for the worst, in the sense that you set your sample size, the maximum sample size, to be, let me see if I can find the slide here that pick your sample size and your funding so that you're covered for the worst case, where you get the least amount of covariate adjustment that you think is likely to occur. So that could just be 0, typically no benefit. In some cases, like the stroke trial example, I told that you might say, all right, I will get at least a 10% improvement. And but I don't want to assume anything beyond that. That's one possibility. So plan that you might not get any benefit, but that can be learned as the trial is progressing. And that's this idea of we call it information adaptive designs, where the information is getting measured over time as more participants are completing the trial and their data is accruing. There you can measure in the trial how much precision gain you're getting, and you can tailor the sample size based on that. So if you're not getting any benefit, then you just go to the end, the maximum, the worst case that you planned. But if you are getting a substantial benefit, then you would be able to stop earlier than you otherwise would. That doesn't always work, especially if you have fast enrollment and the outcome is measured very far in the future, or it's a long period between enrolling someone and measuring their outcome. Then that adaptive information adaptive design isn't possible. But in designs where it's a relatively slow recruitment process, and the outcome is measured in a relatively short duration compared to the enrollment period, then it is possible to do that and essentially try to learn on the fly. That's the best way, I think, to do it, because that's what we propose to do.
Should we take one more live now and then save
Yeah,
some?
I think so, Diane. I think we're going to use the 10-minute time for the audience, the 10-minute time between presentations to run over with questions, and either one or two more questions. And the ones that are not answered, look for answers in the Q&A, and we'll bring them up again at the time of the panel.
And again, I want to make sure we also acknowledge Josh Betz, my colleague from Byrd, Johns Hopkins Byrd, who's also answering questions as we speak. Michael, another one on marginal average treatment effect versus conditional. And so the question is about the packages referenced here allow to calculate marginal treatment effect versus more standard packages provide conditional effect. Is this right?
That's a good question. I would say that the standard, I guess I'm not sure what they mean, the person, well, first of all, the question about marginal treatment effects versus conditional treatment effects is a really good one. My opinion is that both are interesting and useful to learn about. My focus is on marginal treatment effects, but I also think conditional treatment effects are important as well. And so the focus here has been on just methods for estimating marginal treatment effects, and that to get a sense of what that is, a marginal treatment effect, that's what you're estimating with the unadjusted estimator, the difference in means or the difference in proportions between treatment arm and control arm in the primary outcome. So that's what it's estimating, the average treatment effect. What would happen if everyone had been in the study population had been assigned to treatment? What's the average outcome versus had everyone been assigned to control? And some contrast, the difference or the ratio, it could be a risk ratio or something like that. So in terms of estimating, so here are some packages, and actually the longer list is here. In terms of estimating conditional treatment effects, one typically has to make a model assumption, such as assuming a logistic regression model is correctly specified. But if you do make that assumption, then I think you can fit those models. And I would direct questions on that to Frank Harrell, who has much more expertise on conditional effect estimation than I do. So I would defer to him.
We can take this up during the panel at the end as well,
yeah.
Mike.
Absolutely.
Yep, that is great. And I apologize to everyone that I unfortunately have some child duties that will preclude me from being in that panel discussion. But you're probably all better off. You have less, I won't be taking up room from the other great people on this panel. And let me just turn to, and I'll say that the FDA, you can look at their guidance document that I mentioned, the 2023 covariate adjustment guidance document, they talk about both types of marginal and conditional treatment effects in there as both being reasonable choices. And let's see, is
There's
there
one other, I think, very interesting but basic question that I think people would like to hear your answer on. It looks like it may have had a partial answer, and it's how do you determine what is biologically likely important as a baseline factor in picking these covariates?
Yeah, also a great question. I mean, that is one of the challenges is how do you pick the baseline variables in an optimal way or a good way? You don't. So, well, I can tell let me just go back to a particular example that might make it actually, let me do the Alzheimer's disease one that I think illustrates it nicely. So in this is the Alzheimer's disease trial planning example that I used. So the baseline variables here, that's in the middle of the slide here, the age, gender, and then these different assessments that are done at baseline, cognitive assessments, functional assessments, and excuse me, so those were variables or similar to that were selected for the analysis in the trial we were planning and that got conducted. And it has to be decided beforehand. So as I mentioned very early in the talk, this has to be pre-planned. This isn't something that you want to avoid playing around with putting in different combinations of variables to try to game the system or something like that. So let's just pick a set of variables. I tend to be on the more conservative side in the sense of probably picking a relatively small number of baseline variables. The ones that are known both to have a biological connection. So there's some mechanism known connecting the baseline variable to the outcome, or it's just and also the data from previous trials or observational studies shows a correlation, not a tiny correlation. And so ideally you have variables that have both of those. It's also, you might include one if the data from previous trials shows a strong contribution to that there is a strong correlation, even if there is an unknown biological mechanism, and vice versa. If you have but it is tricky how to select the variables because you really should select carefully. If you put in variables that are essentially useless, they don't explain variation in the outcome, then you're wasting a degree of freedom every time you do that. It's not in a large trial that's not terrible, but it's still wasteful. And but I will point out two things that often it's a little confusing at first. Oops. First is that it's okay to include variables that are correlated with each other, baseline variables that are expected to be correlated with each other. For example, these different scores on these different scales, they're typically correlated with each other, but that's okay. That doesn't cause any problem. The worst case would be it's just wasteful if adding one of these, for example, the ADAS cognitive score, if that doesn't provide any additional explanatory value in terms of the primary outcome, given all the other variables that you're adjusting for, if there's no gain in how prognostic these variables are altogether compared to if you left that one out, then it's a waste. But typically the variables each have something that they add. And so there's no problem to put variables that are going to be correlated with each other. Typically they will be correlated with each other, at least to some degree. That's one thing. The second thing is that it's okay. And actually the first variable I think of to adjust for in a clinical trial is if in the case where you have your outcome is a change score. So for example, here, the change in clinical dementia rating sum of boxes score at 15 years versus the baseline assessment. The first variable I think of to adjust for is that assessment at baseline. And there's instinctually, depending how you've been taught about regression analysis, that might not square with you, or you might feel like that's going to screw things up, or it's not okay. In this context, though, we're not just fitting regression models here. It's different than that. So, in almost all in most cases, so that and always it's okay to do that, because that you'd think that might be prognostic, depending how severe you start out, the severity of disease, that could be correlated with your change, and you'd want to be adjust for that. So you don't try to translate what you know about regression models to this context. It's not the same thing. Let's see. Yeah, so maybe that's, I think, a good place to stop.
Right.
Unless I leave it to the organizers. But
Thank you, Michael.
I think we have time for one or two more questions, Guyan. There were follow-up questions from Ronan Fitzpatrick and Navneet Hakua. Ronan is asking you, for ordinal outcome, Wilcoxon statistic can be shown to be a subset of proportional odds model, as per Frank Harrell. Does the usage of marginal ATE affect this interpretation?
Okay, good. So no, I'm happy to answer as many questions as people can ask. And I should mention that I will answer the question, but I'll mention that the reason that I'm excited about covariate adjustment and work a lot on covariate adjustment. I mean, I would be happy to talk all day about it, all the interesting stuff about it. There are two reasons. One is that theoretically you can show the value added from it, but it's not just a theoretical thing. It's actually in real trial data sets. So I've analyzed data from completed clinical trial data sets across a variety of disease areas. I mentioned a couple, but I've also looked at HIV treatment trials, HIV prevention trials, implantable device trials, a wide variety. And in most cases, there is a substantial benefit to covariate adjustment. So it's not, if it were just a 1% benefit in sample size reduction, I would still recommend people use it, because why would you not want to do that? The cost is essentially nothing to get that, but I would not be giving this talk because it wouldn't matter that much. I give the talk because the benefits are sometimes really large, like a 20% reduction in the sample size you need for your trial. I mean, depends on the context, but that could be a lot of money, a lot of patient time saved. So it's more ethical to use it, because you want to minimize the number of participants needed to answer the scientific question in the trial. So that's one of the reasons I'm passionate about it. But going back to the question about the interpretation. So the interpretation in the ordinal case is pretty interesting, because it's tricky. This is getting into what is the estimand. There's an FDA guidance also I'll mention on estimands. That it's a revision guidance and it talks about different possible estimands. But I'll just tell here, for binary outcomes, maybe the simplest type of outcome you could think of, there you might look at the risk difference, the difference in proportions of having a 1 as your outcome, say a modified Rankin scale less than or equal to 3 at 180 days. So that represents a 1 outcome. Otherwise, this is 0. If you take the difference in how many people have this good outcome, a 1, in the treatment arm versus in the control arm, that difference, that's the risk difference, that difference in proportions, you would estimate it that way in the unadjusted analysis. But what you're trying to get is, what if everyone in the study population were assigned to treatment versus everyone assigned to control? What would that difference be? That's what you're interested in. So that's a risk difference. You also might be targeting, you're trying to estimate a relative risk, just a ratio of those probabilities, or an odds ratio, which I would say is the worst option you could choose if you have other choices available. But it's a marginal odds ratio. it's not conditional on any other variables. It's just a number, a summary number. And let's see. And then for ordinal outcomes, here are a few choices. So I skipped over this part, but it's important to say before you even talk about what estimator you're going to use, either unadjusted or covariate adjusted. And if it's covariate adjusted, you have to say exactly what it will be. What is the estimator? What are the variables you're adjusting for? How exactly you're going to do it. In the study, in the statistical analysis plan, that has to be spelled out. And so here it's a tricky one. This came up during at the beginning of COVID pandemic. FDA reached out to a variety of groups and asked for recommendations on estimation methods, but also estimates, because you had scales as WHO scale. There are other scales for different severity of outcome for COVID, and it's a hard question. Is the first thing of what summary difference is the most important one. And so just some examples, you can say the difference in means. You can still do difference in means, but it's interpretation is hard there, because the difference between level 7 and 8 might be completely uncomparable to levels 1 and 2 difference. So that's an issue. There's something called the Mann-Whitney estimand, which is the probability that if you take two people from the study population at random, assign one to treatment and one to control, what's the probability that the treatment person has a better outcome than the control person, assigned person, with ties broken by a coin flip. And so that's the Mann-Whitney estimand. So that does tell you something about, but it doesn't tell you everything. It doesn't tell you how much better if one is doing better. And so there's log odds ratio is a possibility. And so it's challenging to try to condense into some summary what you're trying to estimate. And we have methods for, once you pick your estimand, whatever it is, there are covariate adjusted estimators for it. And let's see. But the nice thing about the interpretation is that there's some issues of collapsibility and what happens to interpretation of treatment effects when they're conditional. While I'd say for the marginal treatment effect, it's just one number, and no matter how you estimate it, the interpretation is unchanged. So that's it's good. It's also, conditional effect is giving you more information. It's telling you what the treatment effect is, not just on average overall, but within strata of different subpopulations, which is, of course, it's something I would want to know, but it's more challenging to estimate it and in general. So it's a trade-off. I'm not sure I actually answered the specific question that was there. So feel free to email me